Hi class, we're going to wrap up the rest of chapter 15, which is our vision and so our eyesight. Let me switch to screen sharing mode. Oh my, I don't see it on here. Let's find it. It's here. Where is it again? Sorry. Keeps getting lost in translation. Here it is. Okay. So this is right where we left off in the last PowerPoint. And these objectives are already posted for you in your chapter folder or your weekly folder in Blackboard. So we'll skip through those. And let's talk about the process of eye development. Just like the brain, we have ventricles that kind of develop and start the brain flowering or blooming that we've talked about already. Eye development starts as little bulges called optic vessels, and they're actually forming from the bases of the brain in development. So your eyes are a neural-based tissue, and as you will see as we move through this chapter, your eyes have a strong neural component to them. So those lateral bulges form optic cups, and they're basically going to be connected to the diencephalon, which is obviously our thalamus and hypothalamus, and different parts will make up the different structures of the eye. Inner and outer layers will develop into the retina, and the neural tissue on the outer layer becomes the basic neurons, ganglions, and special glial cells, which are there for supporting. Finally, the embryonic cells will aggregate around the optic cup, which will basically form all of the supportive connective tissue layers, like the fluids and things like that that are within our eye. This is again showing you that neurocell that's being developed, the optic vesicles on either side, and again how they kind of cup around and envelop this area where the lens is being laid down. So the neural tissue kind of grows up and around. It's kind of an interesting perspective on how the eye is formed. It's kind of a really cool developmental process for people that are interested in developmental biology like I am. These questions here, you can pause the video, go back and review. Let's keep moving with some accessory structures associated with the eye. You have the eyelids and the eyelashes. The eyelashes are very robust hairs, and the whole purpose is to prevent foreign material from going into the eye and irritating it. The eyelids are also called palpebrae. They're a continuation of our skin. Continual blinking helps to keep our eyes moistened and lubricated, as well as to remove any dust or debris that gets in there. And they can also close to protect the eye. Our eyelids have a medial canthus, which is the medial connection between the eyelids, the lateral canthus, which is the lateral connection between the eyelids, and then the fissure, which is the gap between the upper and lower eyelids. That's where our eye is, between that fissure. Other structures that produce secretions are lacrimal caruncle. This is a reddish triangle. That's that little pink triangle in there. It produces thick secretions that can tend to get gritty at night. This is our sleepies that get stuck in our eyes, like little crusties. The tarsal glands or meibomian glands, these are little sebaceous glands that are within the inner margin of our eyelids there, just protecting, just right underneath of our eyelashes, having this kind of fatty, lipid-rich secretion to help prevent our eyelids from sticking together. Other structures that are associated with the eye, what you see in that palpebrae fissure, or palpebral fissure, the cornea, that's the transparent surface, it's the clear layer. The iris is the colored part, the pupil is the black part, or the opening where the light passes through. There, you see what I mean? The palpebrae is the opening where you see the eyelashes. Those meibomian glands would be right in that little surface. Our iris, the cornea is just the clear layer on top, and then the pupil in the middle. The lacrimal caruncle is that little pink piece of tissue. So let's talk about the conjunctiva. The conjunctiva is the surface covering of our eyelids and the outer surface of our eye. So there's a palpebral conjunctiva and an ocular conjunctiva, and the palpebral is covering the inner surface of our eyelids. The ocular is covering the anterior surface of our eye. It's just an extra layer of protection on top of our cornea, and the fornix is the junction of the conjunctiva. So here you see what we're talking about. Our eye lid coming down. Here's our eyelashes, right? And there's this little fornix going in there, and the outer projection is the ocular conjunctiva, and then the projection on the skin is the palpebral conjunctiva. So, and these little white things are just the connective tissue. This is probably actually a tendon from one of the eye muscles. So conjunctivitis or pink eye is the inflammation of that conjunctiva. So the most obvious signs is redness, it's itchy, could be caused by a bacterial or pathogenic infection. It can also be caused by physical stress, allergic stress, chemical irritation. There is a pink eye, looks very uncomfortable. Oh. Other structures of the eye include the layer of fat that surrounds our eye. Our eyes are sitting in a very fatty pocket for padding while the eye is moving around. The lacrimal apparatus, which is made of the lacrimal gland, which is a little almond-shaped gland on the lateral side, versus thick, watery tears. It contains lysozyme to help fight off bacteria. The lacrimal apparatus also includes the tear ducts, which are behind the upper lid, kind of forming like a little lake up there. The puncta, which are little pores that drain the lake, not drown the lake, that would be funny. And the canaliculi are the little canals that are taking those tears and draining them to the lacrimal sac. Now the lacrimal sac is nestled within the orbit itself, and that is the beginning of the nasolacrimal duct. So the nasolacrimal duct is the little passageway from the eye, basically, into the nasal cavity, which is why when you're crying, the ugly cry, right, when you got lots of tears going, you're also going to get a watery nose associated with that. It's just that duct draining into your nasal cavity. Here you see the whole apparatus, the lacrimal gland, the tear ducts going into and behind the eyelid. And as the tears are produced, they're literally washing over the surface of the eye, getting collected at the puncta, draining through the canaliculi into the sac, and then the nasolacrimal duct. So on a normal day when you're not crying, or a normal time when you're not physically producing tears, this is a very passive process, and the tears are just evaporating off the surface of your eye. But when you're producing lots and lots of tears, then it's going to drain and go down into your nose. You can pause the video here, go back and review. We're going to talk about the layers of the eye. We say that the eye has 3 tunics or layers. There's a fibrous layer or a fibrous tunic. This is the outermost layer of the eye. It is consisted of the cornea, which is the clear layer, and the sclera, which is the white layer. It's joined at what's called the corneal limbus, and the overall functions of this fibrous tunic is to support and protect the eye, provide an attachment site for those extrinsic eye muscles that are going to help move our eye around, as well as help with focusing the light into the back of the eye. The vascular tunic, or uvea, is where our blood vessels and lymphatics and smooth muscles related to the eye are. This is going to provide a route for blood vessels to come into the eye, as well as lymphatics. It's also regulating the amount of light entering the eye. It's also secreting and reabsorbing something called aqueous humor, and it controls the shape of the lens. It's also the colored part of the eye. The iris is the colored part of our eye. It contains all those blood vessels. It's very pigmented, and it has layers of smooth muscle which constrict and dilate in response to sympathetic and parasympathetic stimulation. The front surface of our eye is incomplete. It has layers of fibroblasts and melanocytes on that anterior surface of the iris, and then the posterior surface of the iris, the inside part of the eye, has that neural a neural component to it as well. So eye color is determined by genes, and it's also by the density and distribution of melanocytes, which is, again, coded for by your genes. So the density of the pigmented epithelium is basic. You have very few melanocytes in that area, then your eyes are going to be lighter. You have lots and lots of melanocytes, then your eyes are going to be darker. If you have no pigment, then they're going to be a pale gray or a blue-gray color. So other parts of the vascular layer include the ciliary body. The ciliary body is a thickened region that's in the inside anterior cavity. It is made of the ciliary muscle, which is a very small, smooth muscle, and it has the ciliary processes coming off of it, which connect to the ciliary zonial, which hold the suspensory ligaments, which hold the lens in place. You'll see a picture and it'll make a lot more sense once you see how all of this fits together, but it's all part of what's called the ciliary body. The choroid is the vascular layer that is underneath of the sclera. It has lots of capillaries in it, and this is what's delivering oxygen to all of the neural tissue within our eye. The innermost layer of our eye, so again, remember, it's 3 layers. The outer layer is fibrous, then you have this vascular layer where all the blood vessels are and all those little smooth muscles I just mentioned, like the ciliary body and the iris. And then underneath of that, and the inner part, is our retina. This is the innermost layer of the eye, has 2 layers, an outer pigmented and an inner neural layer. That inner neural layer is where our photoreceptors are. These are very sensitive to light. The oda serrata, this is the kind of jagged anterior part of the eye, and this is where the pigmented part kind of begins. So let's put it all together. Here is our outer fibrous layer. You see the cornea and the sclera. The sclera is the white clear part that covers the back of the eye. The cornea is the thickened fibrous part that covers the front of the eye. You have the corneal limbus, which is kind of the boundary between the cornea and the sclera. Then in our vascular layer, this is this kind of pink layer that surrounds the entire eye. That's where all of your blood vessels are. That's feeding the neural tissue. This also has your iris. This right here is the smooth muscle. That's the colored part that you see. And obviously the pupil is the hole in between that. And then the ciliary body is this extra piece of flesh that's holding on to the lens. It has the suspensory ligaments, which hold the lens in place. Oh, and then the inner layer is the retina. So let's talk about the cavities of the eye. The eye has the anterior and posterior cavity. The anterior cavity is divided into the anterior chamber and posterior chamber. So within that anterior cavity, there's even more subdivisions. The anterior cavity is basically from the front of the eye, the cornea, back to the lens. This is filled with an aqueous humor. It's very liquid. It's kind of salty. It's just a little bit more viscous than regular water. The anterior chamber is from the cornea to the iris. The posterior chamber is from the iris to the lens. So it's just a little dividing line there. The posterior cavity is the majority of the eye. So this is everything behind the lens to the back of the retinal wall. This has something called the vitreous humor. The vitreous humor is a very thick, jelly-like substance. So let's look at this cross-section of the eye. You see what I mean? Here's our anterior chamber, this whole purple area, or our anterior cavity, this whole purple area, anterior chamber, and the posterior cavity, which has the vitreous humor. Let's talk about this vitreous humor a little more. It's secreted by special epithelial cells from the ciliary processes. It's very similar in composition to cerebrospinal fluid. It circulates within that anterior cavity, and it actually is going to pass through the pupil to get to the different chambers, anterior and posterior chamber. And it will also diffuse through the vitreous body across the retinal surface. So it circulates all through the eye. Overall, the function of the aqueous humor is to transport nutrients and waste, help to form a liquid cushion or fluid cushion. It also helps to maintain the shape of the eye by kind of filling the space so the eye isn't empty and squishy, you know. And it also helps to stabilize the position of the retina, hold that back and in place so it doesn't shrivel up and implode in the eyeball itself. So the aqueous humor leaves the eye at something called the canal of Schlemm, or as they're calling it now, the scleral venous sinus. I learned it as the canal of Schlemm. It's just this little passageway that goes next to that corneal limbus. So right in that little kind of area in the back of the eye. It flows into the veins in the sclera, and then it's the rate of removal should keep up with the rate of replacement. Tonometry is the procedure that's used to measure the intraocular pressure, and it's usually between 12 and 21 millimeters of mercury. It's relatively low pressure. Here you see this kind of draining. Again, it's created here or secreted at this junction within the ciliary processes, circulates around, drains out of the scleral venous sinus, goes back into the venous circulation, and is cleaned and filtered, etc. You can pause the video here, go back and review these 3 questions. Let's talk about the cornea. The cornea is what allows the light to enter the eye, so it has to be transparent and clear. It has a dense matrix of collagen fibers, which are organized in a way so that light is not impacted by their arrangement, and there are no blood vessels in this area, so it receives oxygen and nutrients only from the tears that are going across the surface. The pupil is allowing for the passage of light into the interior part of the eye. The size of the pupil is controlled by the iris. The dilation of the iris, as I've said, is controlled by the autonomic nervous system, by pupillator dilator muscles and pupillary constrictor muscles. The pupillary dilator muscles are stimulated by the sympathetic nervous system. When you're in sympathetic activation and you're on edge and you're in fight or flight, your pupils are dilated because you're taking it all in. You're nervous, you're scared, you're going to run for your life, right? Pupillary constrictor muscles are your rest and digest. Oh, I just ate a big meal. I want to go take a nap. Your pupils get smaller. Who cares how much light you're letting in? You're going to go rest anyways, right? So it makes your pupil smaller. Here you see what I mean. Pupillary constrictor muscles. There are two layers, and we're about to talk about them. But basically, one is controlled by sympathetic and one is controlled by parasympathetic. So, and the constrictor is the interior, the dilator is the exterior. Same image, just over and over again. All right, let's talk about the lens. The lens is posterior to the cornea. It's held in place by the ciliary body, the ciliary zonule of the ciliary body. And there are concentric layers here. It's filled with transparent crystals called crystallins. And this is basically, those crystallins are responsible for the focusing power of the lens. So it's very dense and fibrous and mostly clear. It's like an opaque color. So again, the primary function of the lens is to focus the light onto the photoreceptors at the back of the eye. And it does this by changing shape. It changes shape in response to those little suspensory ligaments pulling or relaxing to allow it to kind of elongate or shrink up and puff out. So the ciliary body is ultimately the organ in your eye that's responsible for the lens and controlling the lens shape. The retina is where your photoreceptors are. This is pigmented cells, supporting cells, as well as neurons, all within the retina. The photoreceptors are located on the innermost side of the neural portion. There's also something called a macula. The macula, or macula lutea, is the highest concentration of photoreceptors. The central area of this macula area is called the fovea. It's sometimes called the fovea centralis. It's not to be confused with the fovea capitis of the femur. Same idea. It's like an entrance point or a major point of focus. But here we're talking about the eye, not the femur. So this is your point of sharpest visual acuity, or your sharpest vision. So in other words, as I'm lecturing, picture, sitting in an office, my computer's in front of me. That's, where I am. And on top of my computer is this little black thing, right? That's my camera. So I'm looking at the camera, but I can still see everything else in my office. What that means is the light that's coming off of the camera and going into my eye is focusing on that fovea. Everything else that I'm seeing, I can see the pictures of my kids over here. I can see all my to-do lists over here. I can see the PowerPoint and my keyboard all down here. But all of that light is being focused everywhere else in my eye, because I can't see that stuff as detailed as I can see this camera that's sitting in front of me right now. So the fovea is where the light that you want to focus on is going. So let's talk about this idea of a visual axis. It's an imaginary line drawn from the center of any object you're looking at directly through the center of the cornea, through the lens, and onto your retina. So the light would then strike the fovea region on the retina. Exactly what I was just explaining. So there's some other eye structures that are involved. The choroid blood vessels provide nutrients to all of the structures within the eye. The sclera, or the white of the eye, is dense fibrous connective tissue with elastic collagen fibers. It's thickest on the posterior surface and thinnest anterior, because that's where you also have your cornea. It stabilizes the shape and allows for the insertion of all of our extrinsic eye muscles. The optic nerve is also an eye structure. This is cranial nerve number 2, carrying all the visual information back to the brain. Here you see everything put together. You can see the cornea, you can see the sclera, you can even see some extrinsic eye muscles, the retina, the choroid is that layer just deep to the retina, or I should say superficial to the retina, and then the sclera or fibrous layer on the outside, our lens, the ciliary body, and then of course our optic nerve at the very back of the eye. Notice the visual axis on this one image. This is that line of sight going in through the eye, through the retina, or through the lens, and directed all the way back to the retina. The fovea centralis is just off to the side of that optic nerve, and again, that's that point of finest visual acuity. You can pause the video here, go back and review. Let's talk about the process of focusing. The process of focusing involves something called refraction. Refraction happens when light passes through different mediums. So this is why when you look at a fish tank, right, from the side, the fish looks like it's right here. But then when you look at a fish tank from the top, you see the fish was actually right here. So it changes the perspective because the light is bending in different ways when you look through the glass and then look through the water and look through the air, or if you're just looking through the air and the water. So in other words, the more mediums that light has to pass through, the more it's going to bend. And if you think about it, the light has to pass through your cornea, it has to pass through your lens, through the aqueous humor and vitreous humor. So it's bending a little bit all along the way. So your lens has to be super smart and contract or dilate, in other words, elongate or shorten, thicken, in order to refract the light enough to focus it on the fovea centralis. So it can predict and know that. High functioning here. So light is refracted as it moves through all of the parts of the eye. Bending occurs because of the change in density of the medium, because light is an energy wave. So as it moves through a different medium, it's going to slow down or speed up. So let's talk about the focal distance. This is something else that is required to go into focusing of the retina. This is the difference between the center of a lens and its focal point. This is determined by the distance from the object to the lens, as well as the shape of the lens. So cameras basically focus by altering the distance to the lens, right? They're going to move that lens forward or backward in order to focus. In our eyes, the distance from the lens and retina can't be changed. So we have to change the shape of our lens, and that is what we call accommodation. So in this image, you can see the light from a different source needs to be focused on this red dot, which is our focal point, the fovea centralis. Check out the shape of our lens. But when we're looking at something that's very close, right, our lens may have to change shape in order to get all of that light to be focused on the focal point. Or if we're looking at something that's further away, it might need to plump up. And so let's talk about how accommodation happens. It's basically the result of the ciliary muscle action. For close vision, those ciliary muscles have to contract, which means the ciliary body is moving toward the lens, which means there's a reduced tension on the ciliary zonial, which means the elastic capsule of the lens pulls the lens into a more spherical shape. That increases the refractive power, and therefore your lens is now able to focus on nearby objects. As we get older, this ability to pull the lens and stretch the lens decreases, which is why as we get older, most people become more and more farsighted, right? If you know someone that's maybe in their 50s, 60s, 70s, they might look at a sheet of paper and they might have to read it like this, because to read it like this it's too close and they have to stretch out a lot in order to see it, and that's because their lens is no longer flexible. So near point of vision, this is the inner limit of our clear vision. This is determined by the degree of elasticity of our lens. It increases with age, and as the lens becomes less and less elastic, as I was just saying, it's going to reach a limit where it's no longer able to bounce back. So by age 60, our near point is about 33 inches away, which again means you have to really, really hold that sheet of paper or whatever pretty far away in order to read it. So for distant vision, the ciliary muscle relaxes. This causes increased tension on the ciliary zonial, pulling at the circumference of the lens, so the lens becomes flatter, decreasing the refractive power and the ability to focus on distant objects. So here you see, for close vision, this is what's happening. Our lens is getting fatter and focusing on the back. For distant vision, our lens is becoming flatter and able to, again, focus that image on the fovea. So the image is formed not from a single point, but from large numbers of individual points, kind of like the pixels of a computer or of a TV, right? So light from each point is focused on a different part of the retina. And because of refraction, the full image is inverted and reversed. So what you're actually seeing in your eye is a upside down, backwards image of whatever you're actually seeing, which is kind of like, whoa, it's a lot to think about, right? Your brain compensates though, and your brain compensates by reversing the image in that visual cortex. So, and this is through experience that your brain learns to do this. So if what you're actually seeing is this up, right side up telephone pole, what is hitting the back of your retina is an upside down telephone pole. And even notice that that little canister thing is on the other side, whatever that junction box or whatever they're called. Same thing here. This little train track thing, it's inverted and it's upside down. So you can pause the video here, go back and review these 3 questions. Let's talk about the retinal layers and the cells involved in the retinal layers. First, you have that pigmented part of the retina. This is absorbing the light that's passing through the neural part. And this is what you have to understand. When that light comes in, it's passing through the lens, through this vitreous humor, which is very, very thick, and it's just going to bounce around, right? Unless it has something to absorb it. And that's what this pigmented layer is. This pigmented layer is a layer meant to absorb the light. So light comes back and it hits this pigmented layer, this layer that's very darkly pigmented, and it's absorbed, right? The color black absorbs light, the color white reflects light. So this retinal layer in the back of our eye that's pigmented is very dark in color to absorb all of the light coming in. Our cells have very important biochemical interactions with that photoreceptor part, and we're going to learn all about that in a few minutes. So pigmented layer. Then we get to the neural part of the retina. The neural part of our retina has photoreceptors, supporting cells, and neurons. This is going to form the basic preliminary processing and integration of that visual information. Then the outermost layer, which contains all of our photoreceptors. The innermost layer is formed by our ganglion cells, and those ganglion cells basically form the axons of our optic nerve, which go back to a spot of our eye called the optic disc. The optic disc is a functional blind spot for our eye. We have no photoreceptors where that optic disc is. That's where the optic nerve leaves our eye. So there's obviously no photoreceptors there because that's kind of like the hole in the back of our eye. So it's called a blind spot. You can see your blind spot if you go into an unfamiliar area. Let me back up. You would not be able to notice your blind spot in a familiar area because your brain knows what's in that area already and your brain will compensate. That's how smart we are, right? But if you go into an unfamiliar area, next time you're in your lab classroom or next time you're in the library on campus or something like that, you can test your blind spot by literally just using one eye and throw your finger out there, and eventually your finger will disappear. If you move it slowly enough, and that is where your blind spot is. You just do this little finger, move your finger out, and eventually it'll disappear. But again, you have to do it in an area that you're unfamiliar because otherwise your brain compensates and will make it appear even though it's not there. Give you a little optical illusion. Here's what you see. You can see what I mean. Optic disc, there's no photoreceptors here. You can see all these photoreceptors lining the entire back of the eye except for this area where all of these axons are leaving to go back to the brain and within the optic nerve. You can see this back brown layer, that's the pigmented layer. Then you have all of these photoreceptors and then the ganglion cells, which again are all forming together to create that optic nerve. Here is the actual part. You can see this point of highest visual acuity, the fovea there, and then where all this light is, that is our optic disc. It's basically where all of the blood vessels are entering as well in the back of the eye. So let's talk about those retinal cells. The photoreceptors are rods and cones. You may have heard of that before. The rods are highly sensitive. They enable us to see in dim, low lit settings. There's no discrimination against colors in rod vision. It's all just gray, black, and white. Cones provide us with color vision. They give a very clear image and they require more intense light. The rods and cones synapse with the bipolar cells, and the bipolar cells synapse with the ganglion cells. The ganglion cells form the axons of the optic nerve. You also have horizontal and amacrine cells. These facilitate or inhibit communication between the photoreceptors and bipolar cells, or the bipolar cells and ganglion cells. So horizontal cells are the ones that inhibit or facilitate photoreceptor bipolar cell information. Amacrine cells are going to inhibit or facilitate bipolar and ganglion cells. And these will make more sense once you see all of these cells put together. So what I suggest doing is listen to me now and then go and either have that PowerPoint in front of you with the actual picture, or have your book in front of you with the picture of the neural part of the back of the eye, so you can see all these cells, so you can relate exactly what I'm talking about to the actual picture. It's hard to do that on a PowerPoint. Otherwise, I'd only be able to put a few words on every slide if I had a picture for every slide. So I have to break it up some way. Either way, these horizontal and amacrine cells are basically altering the sensitivity of the retina. They play an important role in adjusting to bright or dim light environments. Here's the picture I'm referring to. Pigmented layer in the back. Then you have your rods and cones. Then you have the bipolar cells. The bipolar cells synapse with the ganglion cells, and these ganglion cells, you can see this blue stuff all forming together to make that optic nerve. So the horizontal cells are inhibiting or facilitating communication with the rods and cones and the bipolar, and then the amacrine cells are facilitating or inhibiting communication here. So again, this is all dealing with very dim light or very bright light communication, which is why sometimes when it's dim, it takes a while for your eyes to adjust, and then your eyes can see fine. And when it's very, very bright, you almost like are seeing spots, right? That's because of these two cells that are accommodating. Let's talk about photoreceptor distribution, because it's not equal across the back of the eye. We've already learned about the fovea, which is where a lot of photoreceptors are located, but cones have a maximum density there. There's no rods within that fovea. There's about 6 million cones in each eye, and this is a direct correlation of visual acuity. People that have very, very good vision have very, very good vision because they have more cones and have a higher cone density. Rods have a maximum density on the periphery. There's only about 125 million rods in the eye, so there's way more rods than cones. Rods, again, are just there for to know something's there. They're not giving you a whole lot of information about whatever the object is, but you will know something is there because of your rods. You won't know the colors and everything like that, but you will know the presence of that. Optic disc, fovea on the back. This is showing you visual acuity, again, highest at the fovea. This is showing you density of rods and density of cones. Density of cones is greatest near the fovea. Density of rods is greatest on the periphery. You can pause the video here, go back and review this section. So let's talk about these photoreceptors. They detect photons. Photons are the basic unit of light. Light is measured in waves. Our visible spectrum has a wavelength of somewhere between 400 and 700 nanometers. The visual pigments within our eye are special pigments that are meant to transduce light or break down the wavelengths of light. So they're derivatives of something called rhodopsin. Rhodopsin is made of opsin and retinal. Opsin is a protein that's used to determine the wavelength of a certain pigment, and retinal is a protein that's made from vitamin A. It's a vitamin A derivative. So here's what we mean. This whole thing is a rhodopsin. Rhodopsin is made of retinal, and opsin is this kind of squiggly thing in there. So the photoreceptors have the pigmented epithelium that helps absorb any excess photons that maybe weren't absorbed by the pigmented layer of our retina. They also help to phagocytize anything that's old that may have shed from any other parts that were in that area. There's an outer segment which contains a disc. The disc has the visual pigment in it. In cones, the discs are basically folds of a plasma membrane. The outer segment is kind of blunt and rounded off. In rods, each disc is its own separate thing, and the outer segment forms this elongated cylinder. This is why the cones look like cones and the rods look like rods. They are basically these little discs that are just arranged in different quantities, and they're arranged differently because of their photo processing ability, their ability to deduce light waves. The inner segment, or the inner portion of our rods and cones, is where the organelles are, just like in any cell, right? This is responsible for all the cell functions other than the photoreception. Each photoreceptor is synapsing with a bipolar cell. So here you see what we mean. These are the discs, the connecting stalks, the inner segment, which is where all of the organelles are, and here's the synapse. They synapse with the bipolar cells. So this pigmented epithelium, made of epithelial cells, have these little melanin granules in them to make it pigmented. They have these villi-like projections where the rods and cones are kind of sinking in and being embedded, and this is how they're being activated, right? That light comes in, boom, hits this pigmented layer, and this pigmented layer then can send signals to the rods and cones, and the rods and cones are going to each respond to their own special wavelength, their own special signal. So it's kind of like they have their own language. So let's talk about color vision first. The rods contain all the same type of opsin. They respond to blue-green wavelength as well as white. But there's three types of cones. Cones are, again, very fine acuity. We can differentiate things with cones. You have blue cones, you have green cones, and you have red cones. The majority of your cones are red cones. You do have blue cones and green cones in very small quantities. So combined with all the other stimulation, this is going to allow your brain to determine color. They're all stimulated equally. That's when you see white. So again, it's this kind of orchestra of wavelengths that's going to allow you to see, you know, violet and aqua and pink and, you know, turquoise and seafoam green and, you know, daffodil yellow or pale yellow or, you know, whatever. So it's all based on these wavelengths of light. All of these wavelengths of light are going to trigger the rods and cones at different receptors, which is going to cause them to send different signals. Color blindness is the inability to distinguish certain colors. This occurs when one or more types of cones is not working. This could be present, they may be present, or they may be absent and just unable to function if they are present, unable to manufacture the pigment that they need if they are present, I should say. The most common type of color blindness is red-green color blindness. This is when the red cones are missing, so they're unable to distinguish things like a red light from a green light. But it's a good thing that our red lights and green lights, at least in the United States, all have the same pattern, where red is on top, yellow is in the middle, and green's on the bottom. So, you know, you can see that there is a light, so you know by which one is lit up, if it's red or green. Sex-linked trait is what color blindness is, which means it's on the X chromosome, which means it occurs more commonly in males. Total color blindness is extremely rare, meaning you can see no color. This is a little color blindness test for you. Can you see the number in there? If you cannot, then you have red-green color blindness. It is a number 12, and it's in a reddish-orangish color. You can pause the video here, go back and review this little section. Let's talk about how photoreception is perceived. So first you have the dark. In the resting state, your rods and cones are inactivated in the dark. Chemically gated sodium ion channels in the plasma membrane are kept open in that outer segment. The inner segment is continuously pumping out sodium ions, and this is called a dark current. It's in the dark, makes sense, right? Coming in, going out, coming in, going out. You can picture that image that we saw on that last slide, or maybe you have it in front of you or your book open to that picture, so you can imagine this influx of sodium on that outer segment and then the inner segment pumping it out. This is keeping the resting membrane potential around -40. Remember, every type of cell has a different resting membrane potential. Muscles and neurons were -70. Our photoreceptors are -40. So The photoreceptors are continually releasing neurotransmitters across the synapse to bipolar cells. So that's just something that they're constantly doing. When light is coming into the eye, now the retinal molecule in rhodopsin changes shape. And this process is called activation. It goes from a bent to a more linear shape. That's all you need to know. You don't have to worry about 11 cis or 11 trans. It just means it's kind of flipping forms. Opsin activates something called transduction. This is when a G protein will bind to the membrane disc. If you remember, G proteins then use a second messenger. That's transduction. So transduction will then activate phosphodiesterase, or PDE. Phosphodiesterase breaks down cGMP. Removing cGMP is going to inactivate those sodium channels, and the rate of sodium entry into the cell is going to go down. So think about where we're going with this. Then we create this active state. This decrease in sodium is going to reduce the dark current. So active transport of sodium out of the cell is going to continue. We're going to continue to pump it out, but it's just not coming in anymore. So our membrane potential drops. We get to -70. This is hyperpolarization, right? When we go below our resting membrane potential, that's hyperpolarization. This hyperpolarization decreases the rate of neurotransmitter release. With decreasing signals, to the bipolar cell recognizes that a photon has been absorbed. So that's the little signal to the bipolar cell. It says, there's light, now that we're not getting this neurotransmitter all the time. So here's the whole thing. You have sodium coming in, sodium going out, the dark current, right? The gated channels stay open, sodium's rushing in, and with that, then our transducin is activated. Transducin breaks down GMP to cGMP, and ultimately we're getting that phosphodiesterase, and our sodium channels are closing. When those sodium channels close, does it have it on the next slide? Maybe not. Once those sodium channels close, those synapses are going to become less active. Changes in those sodium ion concentrations are going to hyperpolarize the cell, decrease neurotransmitter release. So rhodopsin cannot respond to another photon until the original shape of retinal is regained. So remember that whole process, retinal was changed from that 11 cis to that 11 trans, right? It changed shape. So there's 3 steps to getting the retinal back. Remember, this is all to see 1 photon of light. All of this happens. It's crazy. Their first step is called bleaching. The entire rhodopsin molecule is broken down into retinal and opsin. The retinal is converted back into its cis shape, and that requires ATP energy. And then the reassembly occurs where the optin and retinal are reassembled as rhodopsin. So here you see this bleaching. You've got to take the retinol out. You have to convert it using ATP and then stick it back in. This whole process of reassembly. You can pause the video here, go back and review. So let's talk about this visual pathway. Do I say that a lot? Let's talk about. I like to talk. Sorry, that's why I teach. So visual pathways begin at the photoreceptors, pass information to the bipolar cells, then to the ganglion cells. Axons from about 1 million ganglion cells convert to the optic disc, proceed toward the diencephalon as the optic nerve, cranial nerve 2, and then 2 optic nerves, 1 from each eye, reach the diencephalon at a point called the optic chiasma, or optic chiasm. It's the same thing. From that optic chiasm, along the optic tracts, about half of them are going to pass the other way. So half go to the lateral geniculate nucleus, the other half go to lateral geniculate nucleus on the opposite side. So half from each eye goes to each side of that thalamus. There's also something called optic radiation. This is a collection of projection fibers which carry information from each lateral geniculate nucleus to the occipital cortex of the cerebral hemisphere on that same side. So taking it back to that visual cortex where that visual image can be perceived and information can be gathered about that stimulus. So right and left eye, information coming back, again crossing over, 50% goes this way, 50% goes that way, and then these projection fibers sending that information back to the primary visual cortex. How do we see depth? Well, the ability to judge depth and distance is by interpreting a 3-dimensional relationship of all of the objects that we see. So it's perceived by comparing the relative positions of objects with images from both sides of the eye. So really, if you just have one eye, your depth perception disappears. Again, if you're in a familiar environment, your brain compensates. But if you're in an unfamiliar environment, your depth perception may really be out of whack if you try to go around with just one eye. Visual images from the right and left eye overlap. That's called binocular vision. Each eye receives slightly different image because the fovea are spread apart and the nose and the eye socket can block views from the other side. So that is how we are able to have that depth perception. You can pause the video here, go back and review. We're going to continue on by talking about problems with refraction. This can result from abnormalities within the lens or the cornea or the overall shape of the eye. Emmetropia is normal vision. This is when the distant image is focused on the retinal surface, ciliary muscles are relaxed and flattened. Normal vision going on. Emmetropia. But myopia is nearsightedness. This is when the focal distance is too short, so the image is actually focusing in front of the retina. This can be caused because the eyeball's too deep or because our curvature of the lens is too great. In other words, it's not flattening enough. It's corrected with a concave lens in front of the eyes, which will help spread the light rays apart to focus that light on the retina. Here you see that diverging lens. So our actual lens doesn't move or change shape, but by providing an extra refracting site in front of the eye, we're now able to focus that image on that fovea. Hyperopnia is farsightedness. This is when the focal distance is too long, so the image is actually being focused behind the retina. This can be caused if the eyeball is too shallow or if the lens is too flat. So it's corrected with a convex lens in front of the eye. So again, just providing additional refraction by having that lens in front of the eye. Surgical correction of refraction problems can be done through something called PRK, or photorefractive keratektomy. This is a computer-guided laser surgery, basically reshaping the cornea, or LASIK, laser-assisted in situ keratomyolysis. This is when the interior part of the cornea is reshaped and recovered by corneal epithelium. 70% of LASIK patients achieve normal vision, high success rate, but I don't know if I would want a laser going in my eyeball. I was blessed with perfect vision, but I don't know about that. That looks pretty intense to me. But it's awesome for people that have had it, who have had to wear glasses their entire life and now have a normal life without vision problems. So congrats if that is you. You can pause the video here, go back and review these 3 questions. We're going to talk about disorders now, going all the way back to our first sense, which was olfaction. So olfaction disorders can result from head injury or any age-related issues where our just receptors become less and less sensitive, right? Or gustation disorders, which could evolve from or develop from olfactory disorders as well, or there could be damage to taste buds from some type of inflammation or mouth infection. There could also be cranial nerve damage or general age-related changes where our taste receptors just become less sensitive. Any condition that affects our sense of smell is also going to affect our taste, which is why sometimes when you're sick with a head cold and your nose is stuffy, things just don't taste as good as they used to. There's also vision disorders like cataracts. Cataracts is a condition where the lens is losing its transparency. This can result from injury, radiation, reaction to drugs. It can also just be a normal part of aging. It's the most common form of cataracts. The damaged lens can be replaced with a synthetic lens, interestingly enough, and the vision can also be refined with glasses or contact lenses. Here you see an eye with a cataract and a normal eye. Vertigo is an equilibrium disorder. This is the illusion of movement even when you're not. It's usually caused by conditions that alter the function of the inner ear complex, the vestibular branch of the cochlear nerve, or the sensory nuclei within the central nervous system. It can be stimulated by anything that sets the endolymph in motion. So motion sickness is the most common cause. There's drugs to kind of depress the activity of the brainstem. Other causes would be like alcohol consumption or exposure to certain drugs, which is where you kind of lose your balance and you feel like you're moving, or you're laying in bed and you feel like the room is spinning even if it's not. Just like that. Equilibrium changes with age, just like everything else. There's also hearing disorders. Partial hearing deficit will affect nearly 6 million Americans. Conductive deafness is an interference with the transfer of vibrations from the tympanic membrane to the oval window. So this would be if there's like too much earwax trapped in the external ear, or maybe some water, scarring of the tympanic membrane, or if your ear ossicles are immobilized by some way. There's also nerve deafness. This is a problem with the cochlea or any part of the auditory pathway. The vibrations of the oval window are there, but the receptors just don't transfer that information. This could be caused by damage to the receptors, like if you listen to your music too loud or to the TV too loud, etc., or bacterial and viral infections can actually kill those receptor cells as well. As with everything, our hearing changes with age. Young children have the greatest hearing range, and with age, damage from loud noises and injuries accumulates. The tympanic membrane gets less flexible, and articulations between our inner ear ossicles are going to stiffen, so they're not going to move as well as they used to. That round window may also begin to ossify, which makes it less sensitive, and obviously all of that results in some degree of hearing loss. Good thing we have hearing aids to help our elderly and individuals that have hearing loss. This is the last slide of chapter 15. You can answer these 3 questions. Let me know if you have questions, and hopefully you're having a good day and this chapter makes sense to you. If not, let's chat, let's talk about it, and I hope that you, I don't know what I was going to say. I hope you enjoyed my lecture. Have an awesome day, and I will talk to you later. Bye.